So chapter 12 focuses on simple Mendelian genetics. And Gregor Mendel, he was a monk who lived in the 19th century. And this is before anybody knew anything about chromosomes or genes or even what DNA looked like. But yet he was able to determine that, or he was able to identify what genes were and how they were inherited. And the way he did this is through simple genetic crosses. And so today we're going to focus on the different experiments that he did and also his conclusions that he found. And so the way that Mendel did this experimentation was with pea plants. And he actually took two different types of pea plants. He had one that was produced purple flowers and one that produced white flowers. And then he crossed them together in order to see what the offspring would look like. And so these white flowers and these purple flowers, they're called true breeding. Because if you crossed a white flower to a white flower, you're going to get a white flower. Just like if you crossed a purple flower to a purple flower, you would get a purple flower. So that's the definition of true breeding. It means if you cross it by itself, you're going to end up getting the same exact phenotype. And remember, we talked about the difference between genotype and phenotype. Your genotype is what your genes are actually coding for. And the phenotype is what the organism is going to look like as a result of that genotype. And so the way that Mendel set up these crosses, he did a series of crosses. For each one of these genetic crosses, you're going to have a parental cross. So that's going to be the first cross. That's the P generation, standing for parental. In this case, Mendel took a purple flower and he crossed it by a white flower. Or I should say pea plant. And then he took the results of that cross, the offspring that were produced from this are called the F1 generation. So the F1 generation is going to be the progeny or the offspring of the parental cross. And so what Mendel saw was he saw all purple plants in his F1. Then what he did is he took two of these F1s and he crossed an F1 times an F1, so a purple by a purple, and he saw something interesting. He actually ended up getting offspring in the F2 generation. So the F2 is the next generation after F1, and the next generation would be F3, and so forth and so on. But in the F2 generation, what he saw when he crossed these F1s to F1s, he saw 3 purple flowers and 1 white flower. And so this kind of struck him as odd because he was expecting to see only purple, because that's what he saw in the result of his first cross. So and when he crosses the purple and the white, he gets all purple. But then when he crosses that purple to that purple, he gets some white thrown in there. And so this led him to think that maybe there were various different traits that were contributing to an organism's genotype. And so he continued to do these different types of experiments. And the way that he would actually do them is he would cut open the male and female parts of each of the pea plants, and then he would actually fertilize the female with pollen from the male. And so that's how he crossed his experiments, and then you let them grow up like normal. And so here you can see after he fertilized them and plant them, then you can see the F1 generation is that all purple flowers, or that cross between those parental purple and parental white flowers. And so the results, as we mentioned, he saw after his parental crossing, these are the true breeding, so he crossed the parent times the parent, and he got 100% purple. And then he took these F1s and he crossed them to one another. So an F1 times an F1, and in the F2, he saw a ratio of 3 to 1. So for every 3 purple flowers, there was 1 white flower. And so he did this experiment several different times, and he came to the conclusion that this purple trait, because it shows up more often than the white one, that this purple trait is going to be dominant to the white trait. And so he assigned these names. The traits that are going to show up more often, are going to be more common, are called dominant traits. And then those that show up in lesser number are going to be the recessive traits. So in this case, he determined that purple, the trait that makes this flower purple, is going to be dominant over the trait that makes this flower white. And so these traits that we're talking about, they're now understood to be genes. Like I said, when Mendel was around, we didn't know anything about chromosomes or gene or DNA structure, so he figured all this out on his own. And it turns out he was correct. So when we're talking about these traits, we're essentially talking about genes. So in other words, the purple flower gene is going to be dominant to the white flower gene, which is recessive. And so he continued to do these experiments, and he did it looking at several different phenotypes. And in all of these tests that he did, he took two traits that were opposite of one another, just like the purple and the white. He also looked at how the flowers were arranged, whether they were at the top of the stem or whether they were on the side of the stem. He also looked at seed color, yellow and green. Also the seed shape, so some are round, some are wrinkled. So all of these different characteristics he examined doing the same sort of crossing or fertilization. And in every single one of them, he found this 3-to-1 ratio. So 3-to-1 purple to white, 3-to-1 axial to terminal, yellow to green, 3-to-1. And so if you look at all these ratios, they're all right about 3-to-1. And so he kind of used this information in order to determine that, or in order to solidify his hypothesis that some traits were more dominant compared to other ones. And so in a simple genetic cross like this, where you're just comparing one gene but two different versions of that gene, that's called a monohybrid cross. So a monohybrid cross is going to be when you're looking at one gene. So in this case, we're looking at the gene for flower color. And so this is an example of a monohybrid cross. And more particularly, it's whenever you cross the F1s to one another in order to get the F2s. That's what a monohybrid cross is. And so, again, he was able to show that there was a 3-to-1 ratio, a 3-to-1 phenotypic ratio observed within this F2 generation. So 3/4 of all the offspring showed the dominant trait, and only 1/4 showed the recessive trait. And so this led him to form several different conclusions based on these initial experiments. The first one that he concluded was that for each trait, and remember a trait is a gene, for each trait there's going to be two versions, all right? Just like for the flower, there was purple and there was white. And so he called these different versions of a gene alleles. And so if I have brown hair, that means one of my genes is at least have a brown allele. My other gene, the one from my dad, could be blonde, but since brown is dominant to blonde, I turned out with brown hair. Same thing with eye color. You have the blue eye color allele, you have the green eye color allele, you have the brown eye color allele. So alleles it's the same gene. The gene is eye color. There's just different versions of it. Just like the gene for flower color, it's just one gene, but you can have alternative versions. And those alternative versions are alleles. And so from his research, Mendel concluded that each trait had different versions. And so if you look at this, these are going to be homologous chromosomes. So one from mom, one from dad. And so each one of these alleles are always going to be localized to a particular region of the chromosome. And that region of the chromosome is called a locus, or a loci. L-O-C-I is going to be plural. But a locus is where you find all of these different genes. And so, for instance, if you look at these two homologous chromosomes, here would be the allele for purple flowers on mom's chromosome, and here's the allele for white flowers on dad's chromosome. So it's the exact same gene, and they're located in the exact same location on the chromosome. It's just you have different versions of those genes. And so this, he came up with this notion based on the idea that some of these alleles were more dominant to others. And so from this, he was able to determine that there were multiple different versions of genes that were contributing to the phenotype in his crosses. The next thing he was able to figure out is that for each one of these traits, an organism is going to inherit both alleles of it, or multiple alleles of it. So just like in the previous example, we showed homologous chromosomes that have, an allele for white and an allele for purple flowers. Well, that plant that's going to arise from that is going to show, or is going to contain both of those alleles. In other words, both versions of the gene. And then what dictates what the phenotype is going to be is which of those traits is dominant over the other. Or maybe you have both the same type of alleles in both of your chromosomes. And so he determined that both of these traits are being inherited together. And then based on the relationship of those traits, whether you have a dominant one or a recessive one, that both of those traits were going to dictate what the offspring look like. And so when we're talking about these alleles, so you can have, as we mentioned, dominant alleles and you can have recessive alleles. If you have an allele that is dominant, we always write it as a capital letter. And so let's just use the example of the flower color. So we know that purple is going to be dominant over white, right? And so we're going to use just an abbreviation to mark that trait. Big P, capital P, stands for purple because it's dominant. And so the recessive allele, it's not going to be a W, it's going to be a lowercase purple. So that tells you that that lowercase p stands for white. And that it's going to be the recessive trait, which is why it's lowercase. So you take usually the first letter of the dominant trait to represent the dominant allele, and then the lowercase letter of that to represent the recessive allele. And so there are different arrangements of these two alleles. The second thing he concluded was that two versions of that gene are going to be inherited together. And then the relationship of the two is what's going to dictate what the offspring is going to look like. And so if you have two different alleles and both of them have the dominant trait, so in other words, the two genes are big P, we call this homozygous. Homo means the same, and so if you have a homozygous genotype, then that means you've either got capital P, or what's the other option? Small p, right? So as long as both alleles are the exact same, it's going to be considered homozygous. Now you can have a homozygous dominant, which is the capital P, or you can have homozygous recessive, which is the little p. And so there's also another term, which is basically a combination between the two, which is called heterozygous. Hetero means different. And so if you have one dominant allele and one recessive allele, we call that heterozygous. And so the three different ways that we can characterize our genetics are heterozygous, homozygous dominant, or homozygous recessive. And so we're going to be using those terms throughout the rest of the lecture. All right, so the third thing that Mendel concluded from his experiments was that the dominant allele, so if you have these two alleles of a gene, and we already know that they're both inherited together, so if there is a dominant allele, it's going to automatically show the phenotype of that dominant allele. So for instance, in the cross that we were talking about, whenever we crossed the true breeding purple to the true breeding white in the parental generation, and we got the F1, all of the F1s, 100% of them were purple. And so that told him that the purple trait is dominant to the white trait. So the dominant version of the gene is going to take over. And then in the recessive trait, you're not going to see any phenotype. And so this is what he observed in that F1 generation. And so this also led him to form the law of segregation. And so he's already concluded that there are multiple versions of genes. And he's also concluded that these genes or these alleles will be inherited together. And now he creates this law of segregation. And the law of segregation says that during meiosis, remember meiosis, we're making the gametes. The law of segregation says that during meiosis, those different alleles or those different versions of the genes are going to be going into different gametes. Because remember, we're starting off with genes from, or a chromosome, one from mom, one from dad. And so during meiosis, we're generating these haploid cells, and so we have to separate those homologous chromosomes from one another. So when we separate those homologous chromosomes from one another in metaphase 1 of meiosis 1, we're essentially separating those two different versions of that alleles. So another way to say this is that when the two alleles of a gene are separated during meiosis, they're going to end up in different gametes. And so if you look back at, so here we have, here's our homologous chromosomes. So in meiosis 1, again, we're separating those homologous chromosomes. So a trait right here and a trait right here, they're eventually going to be separated into different gametes at the end of meiosis. And so from this law of segregation, he determined that the arrangement of the chromosomes in meiosis 1 is going to help dictate the way that the alleles segregate into the haploid daughter cells at the end of meiosis. And so remember we talked about that the chromosomal arrangement at metaphase 1 in meiosis is random. And so depending on the way that those chromosomes align on the metaphase plate is going to dictate how they're going to be separated in metaphase 1 of meiosis 1. And so depending on how those homologous chromosomes are arranged, whether it's, all of mom's chromosomes on one half and all of dad's on the other, or vice versa, or a mix, that's going to dictate how these gametes are going to be formed, and specifically those alleles that those gametes are going to contain. Questions? Have y'all had some basic genetic stuff before? Okay. Okay. So, also from Mendel's studies came about the idea of a Punnett square. And a Punnett square is just a diagram that can be used to predict the offspring of a cross. And so you do have to know the genotype of the parents though. And so in the example of Mendel's cross, we know he crossed a purple flower by a white flower. And when we said that those flowers were true breeding, another word to describe true breeding is that the purple is going to be homozygous dominant because it's the one that showed up more often. And the white one is going to be little p, so it's still going to be homozygous. So anything that is true breeding is going to be homozygous. It's either going to be true breeding for the dominant trait or it's going to be true breeding for the recessive trait. And so another way to say true breeding is just homozygous. It can either be homozygous dominant or it can be homozygous recessive. Anyways, in this first cross, if you put the alleles, I'm sorry, not the alleles, but if you take the gametes that are possible, that are going to be made from this individual, and you cross those gametes to the gametes that are possible in this parent, then you can come up with a number or a fraction or a ratio to predict what the offspring are going to look like. So in other words, we know that this plant right here is homozygous dominant. It's capital P, capital P. So in meiosis, you're only going to make two possible gametes. You're going to have a purple, a capital P, and a capital P. And so each one of these capital P's will be a possible gamete. Same thing for this guy. We know that since he's the homozygous recessive, he's going to be little p, little p. And so when his gametes form, those two alleles are going to segregate. And so all we do is we're crossing the various different possible gametes to one another. So if you just multiply a big P times a little p, you're going to end up with a big P, little p. Big P, little p is heterozygous. And he noticed that for every single possible genetic combination. And so all of these are heterozygous, meaning they have one of the dominant allele and one of the recessive allele. But because that dominant allele is there, those flowers are going to show the dominant phenotype, which is going to be purple. So this is why he saw 100% of the purple flowers in the F1 generation. Then what he did is he took these heterozygous F1s that were a result of this first cross, and he crossed them to one another. So in other words, a heterozygous purple times a heterozygous purple. And so when you look at the way that the alleles segregate during meiosis, you can either have a big P gamete or a little p gamete. And same thing for this guy. You can have a capital P, so a dominant allele here, and then the recessive allele here. So all you do is you look at the